Let's talk about the cell cycle and mitosis. Before we do, however, let's talk about the two types of cell division: mitosis, which is PMAT, and meiosis, which goes through those same steps twice. Mitosis is for growth, maintenance, and repair. Meiosis is for gamete formation, to make sperm and egg. In mitosis, we start out with a 2N cell and end up with more 2N cells. In other words, we start out with 46 chromosomes and end up with 46 chromosomes. In meiosis, we start out with a 2N cell and end up with haploid cells, 1N. Or we start with 46 chromosomes and end up with 23 chromosomes. In mitosis, two diploid cells form. So we start out with a diploid cell. It divides to produce two daughter cells, which are also diploid. In meiosis, 4 haploid cells form. We start out with 2N, which divides once to produce 2 haploid cells, and divides once more, in the end forming 4 haploid cells. Mitosis occurs in somatic cells, whereas meiosis occurs only in gonads, the ovaries and testes, and it is the cause of most genetic variation. The cell cycle has 3 phases: interphase, mitosis, and cytokinesis. Its longest phase, indicated by the longest arrow, is interphase. Interphase also has 3 parts, G1S and G2. During G1, growth and protein synthesis occurs. During S phase, DNA synthesis occurs, which means it changes from a single chromosome into a duplicated chromosome. In G2, more growth and protein synthesis occurs. When the cell is grown too big for nutrients and oxygen to reach the nucleus, then it has to divide, and that is mitosis. Mitosis consists of 4 parts: prophase, metaphase, anaphase, and telophase, and it is when the chromosomes divide. So we have a duplicated chromosome, turns back into 2 single chromosomes. Last, at the end of the chromosomes dividing, is the cell dividing. Cytokinesis is when the cell divides into 2. Some cells don't continue in the cell cycle. An example of this is red blood cells. They don't have a nucleus, and so they cannot divide. DNA replication is necessary so the chromosome numbers stay the same, or to have cell division. If you don't first duplicate the DNA, there's no way to divide a chromosome in half. Interphase is the stage prior to mitosis. Chromosomes are not visible because DNA is in the form of chromatin. Main events are G1, where growth and protein synthesis occurs, S phase, when DNA replicates, and G2, more growth and protein synthesis. Let's label this cell. The cell on your page is just to the right of the box labeled interphase. We have the cell membrane, nucleus, nucleolus. The DNA is in the form of chromatin. And we have 2 sets of centrioles because the cell is getting ready to divide. All the other organelles would be inside the cell. We are only drawing the components necessary for cell division. Prophase, the first part of mitosis. Chromatin becomes visible chromosomes, which look like X's. The nuclear membrane disappears, the centrioles separate, the spinofibers and asters form. This is a view under a microscope of prophase. So we have the centrioles. They send out microtubules called asters to anchor them to the poles of the cells. Next, we have the nuclear membrane disappearing. That's why it's a dotted line. The chromatin has coiled up into chromosomes. We are drawing a cell with 4 chromosomes in it, 2 copies of each, 2 copies of chromosome 1, 2 copies of chromosome 2. Remember that these are duplicated chromosomes. The centrioles also shoot microtubules across, and these are called spindle fibers. This cell has a ploidy of 2n equals 4. There are 2 of each chromosome, and there's a total of 4 chromosomes in the cell. Chromosomes during prophase and metaphase are duplicated chromosomes. Each half is called a sister chromatid, and the part that holds the 2 sides together and joins it to the spindle fiber is called a centromere. Dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. Biology humor. Okay, metaphase is when the chromosomes line up in the middle. The individual's chromosomes can be seen, and here's a picture under a microscope. So let's again draw the spindle fibers. This time the chromosomes are attached to the spindle fibers by their centromere, and they are lined up on the imaginary line called the metaphase plate. The ploidy of this cell is still 2n equals 4. Next, anaphase. We're going to remember A for apart. The centromeres divide and the chromosomes move to opposite poles. This is called segregation. Here we see it under a microscope. And let's draw it. So the reason the chromosomes are separating is because the spindle fibers are being pulled back in by the centrioles. So it pulls the chromosomes apart at their centromere. And now, instead of being called sister chromatids, they are no longer joined, so they are called chromosomes. The ploidy of this cell is 2n equals 4. Telophase. It's about to tear into two, so it's getting ready for that. The chromosomes reach the opposite poles. They begin to lengthen out and again become chromatin. Spindle fibers dissolve, and the nuclear membrane reappears. So here's a picture under a microscope of it. Let's draw it. Here's the nuclear membrane reappearing. We're drawing the chromosomes, so this is early telophase, before the chromosomes have decondensed and formed chromatin. 4 on each side. We can tell it's telophase because of the cleavage furrow. That's what we're going to look for to identify this phase of mitosis. Each side is 2n equals 4. Okay, finally, the cell divides into two identical cells. Okay, so we're going to draw those centrioles again. We've got the nucleus now, and the DNA has decondensed into chromatin. Each new daughter cell has a ploidy of 2n equals 4. Okay, there's one difference between plant cells and animal cells. Cytoplasm pinches in or invaginates in animal cells, so it forms the cleavage furrow. A cell plate is formed in plant cells. This cell plate, which we're drawing here with a dotted line, eventually becomes a cell wall made of cellulose. Animal cells do not have cell walls.